And we are recording almost now. Not recording yet. Okay, the website, though, while it's getting ready, is Arabidopsis.org. Arabidopsis.org. Here we go. We're recording. So this is question 5 of analysis in DSAP. It's the Arabidopsis database question. And so you go to the TAIR database. How do you get there? It's Arabidopsis.org, Arabidopsis.org. And then it'll come up as such. It's the TAIR database. So what Arabidopsis is a tiny little cute flowering plant that the genome was sequenced in the 1990s. It's just a model organism. It's a tiny, cheap, very cute, plentiful, hardy, tiny plant that geneticists have years ago. And so we have this huge genome, the whole thing done, and we're just comparing other cDNAs and parts of genomes to it. It's just a known genome. It's called Arabidopsis.org. It's a very popular organism to research upon. So let's do this. Here's how you do it. You go and get your protein, and I'll get it from Blast. I'll get it from Toolbox. So I copy the protein from Toolbox. Hope that's not too noisy. And we'll go to Tools Blast. Tools Blast. Now you can get your protein, if you don't have Toolbox open, you can get your protein from BLAST-P, Define Orphan BLAST-P of DSAP. It's the same thing, but make sure it's protein, not DNA. So then you click on BLAST-P and paste your protein in. So once again, you're changing that to protein, to amino acids, and you're putting your protein here, okay? Make sure it's protein. And you get the protein from Toolbox or from Define or BLAST-P in DSAP. Okay, and so then you click Run Blast, I think. Yep, Run Blast. And we're gonna send this out and see what our protein matches to. So this is our protein matching to other proteins specifically in this database, specifically in Arabidopsis, which is that thale crest, that little plant we've mentioned. So these proteins are referenced in 2 places, up here in these nice pretty colors, and also down here. It's just a little easier to see, I think. And what you want are these AT numbers. I'm pretty sure A stands for Arabidopsis and T for TAIR. It's telling us Arabidopsis TAIR database, all these are proteins. So let's take one. We're going to take the first one, AT. Copy that. And you're going to put that in your answer for DSAP. Okay. You could take any of these. I'll just take the first one, nice and easy. The next question says something to the effect of, what protein did you use to search the Interactome database? Well, here's the oh, that's not it. The Interactome database is this. Okay, it's the Salk Interactome database. You can Google Signal Salk Interactome database, or you just use the link in DSAP. Salk Interactome database. And you're going to paste in the AT number upper right. And make sure this works with Flash. So you have to click here and make sure Flash is allowed. I'll just reload it. Okay, so I put the AT number in the go box, I press go, and I should get results. And it should look like a flower, let's see. And there it is. And these are fun to play with, see? You can stretch them. So what am I looking at? The one in the middle is that protein from the Arabidopsis database. That's this protein from the Arabidopsis database. Oops, wrong one. So this AT protein in the Arabidopsis database is this one in the middle. Okay, so it's telling me my protein interacts with all of 5 of these. Because proteins are social, so to speak. They don't always just exist alone, or I shouldn't say exist, but they don't always just catalyze reactions alone. They're usually touching a membrane, maybe other proteins. They can ligand with different signal peptides. Sometimes, for example, collagen is quaternary. It's got multiple parts to it. Hemoglobin's got multiple parts. Or it could be an enzyme that it needs another protein to ligand with and stick to, and therefore work. There's lots of reasons why proteins would be interacting with other proteins. And this is just research that people have done that found their protein interacting with other proteins. Looks like a person dancing. So the middle one is a protein, is your protein, and these are all interacting. Choose any one you want. All right, I'll choose this one at 10 o'clock. It doesn't matter. Choose anyone you want. And then go to the right and just scroll down, and there, right here, this has got a cool name, tetratricopeptide. Tetratricopeptide. You can copy paste this, that's the function. Or you can reword it. If you copy paste it, just put it in quotes, otherwise you can reword it. All right, I'm gonna stop recording. That is the TAIR Interactome database.